Hello, my name is Bryan, and along with my group mates, Ting and Jhee, we will be presenting on degenerative protein deamidation. Deamidation is one of the several post-translational modifications, PTMs, which is attributed to many diseases, namely neurodegenerative disorders such as Salix disease, Alzheimer's disease, Parkinson's disease, cancer, and cataracts. Research and interest in the study of neurodegenerative diseases started from the pathology of Alzheimer's in the 1800s. Although the causes of the disease was linked to the brain, physicians of the era could not identify the exact molecular composition of the neurodegenerative disease. In 1854, German physician Rudolf Virchow used histochemical methods available at the time to identify wax-like plaques in the brain tissues from autopsied individuals. He concluded with a paper and a sketch that the plaques were composed of starch. In 1861, Friedrich and Kekule suggested that the brain amyloid plaques were in fact composed of proteins rather than carbohydrates, and in so doing, they established for the first time the presence of protein dysfunctions in the brain. Early in the 20th century, the generalized use of Congo red stain allowed the description of fibril composition of amyloid plaques as revealed by the contrasting red and the green tones exhibited by stain samples when visualized under polarized optics. The presence of protein dysfunctions have been described in almost all neurodegenerative disorders, including Alzheimer's disease, Parkinson's disease, Huntington's disease, and amyotrophic lateral sclerosis (ALS). To name a few, amyloid beta peptide and the microtubule-associated protein tau are the key constituents of brain plaques and tangles in Alzheimer's disease patients. However, some of these mechanisms are poorly understood. Initial hypothesis that protein oligomers initiate proteinopathies has for many years shaped the overall direction of research in neurodegenerative pathologies. However, recent data have indicated that diverse range of soluble proteins can become dysfunctional and exert toxic effects in brain tissues that surpass those of oligomerized proteins. The most recent technical advances in mass spectrometry-based proteomics have now provided us with the necessary tools to uncover the molecular basis of protein dysfunction in diseases of neurodegeneration. The mass accuracy and databases can identify post-translational modifications by their molecular weight on a particular peptide. Deamidation, like all other PTMs, can be identified via reduction, alkylation, and tryptic digestion of the protein, and subsequent HPLC separation of the tryptic peptides prior to mass spectrometric analysis. Post-translational modifications regulate protein functions. In healthy cells and tissues, PTM is induced by enzymatic modifications to the side chains of proteins. PTMs associated with spontaneous chemical processes are often associated with disease progression and hence termed as degenerative protein modifications, DPMs. In some proteins in the tissue cells are long-lived proteins and they are least expressed in the cells. Therefore, the loss of function in these proteins naturally occurs with age. Hence, they are called degenerative proteins. The process of degeneration of proteins are further enhanced by post-translational modifications. Spontaneous processes can cause change in the 3D structure of the protein, can also cause change in important amino acids crucial in regulation, and also can cause bond cleavages in peptide chains, result in mostly irreversible damage, and will affect downstream signals, as well as protein-protein interactions. DPMs are caused by processes namely oxidation, nitration, carbonylation, carbamylation, and glycation. However, this presentation we shall focus on deamidation. Deamidation as a molecular clock was proposed by Robinson in a paper called Molecular Clocks. The paper concluded with 3 key points. First, These proteins that undergo deamidation serve as a versatile clocks for the timing of specific biological events. These proteins are available for suitable timings of events throughout the useful range of the biological processes, as well as to be removed after it has completed its process, so that it does not cause harm to the biological systems. Next, amides can be used as molecular indicators of protein integrity. So if the molecule is altered by deamidation, it can set off a catabolic process of correcting itself to repair itself, as well as to form a new protein function. Lastly, deamidation proteins have a specific function in homeostasis. Non-enzymatic deamidation of asparagine has far been shown to regulate 3 types of processes, including the timing of turnover rates of cytochrome C4 and aldose 5, the counting of individual enzyme catalytic cycles in triose phosphate isomerase 6, and the time-dependent monitoring of DNA repair and the regulating of apoptosis in BCL-XL. 7-asparagine deamidation has been observed in more than 200 types of biological peptides and proteins. Deamidation affects asparagine and in some cases glutamate residues, as these amino acids are unstable in physiological conditions. Glutamate residues undergo slower deamidation than asparagine residues. Asparagine is converted into a racemic mixture of isoaspartate and aspartate. Deamidation on asparagine can occur in proteins and peptides. The mechanic studies have suggested that the reaction is at neutral pH and is intramolecular and proceeds in a 5-membered ring succinimide intermediate. This intermediate step is formed via a nucleophilic attack on the side chain of carbamoyl carbon on asparagine by the backbone nitrogen of the ensuing amino acid residue. This causes a release in NH3. It is believed that the backbone amide is deprotonated prior to the nucleophilic attack. The succinimide ring is then liable to nucleophilic attack by water, resulting in 2 isomers, isoaspartate and aspartic acid. The occurrence of spontaneous non-enzymatic deamidation can be caused by a variety of pathways, which can be dependent on the cellular environment. It can also depend on solvents interacting as catalysis, or it can take a stepwise, least energetic pathway forming tautomers.
Proteins in which deamidation has been established and the site characterized. Glycine is the most common N+1 neighboring residue in 70 to 80% of proteins with asparagine to glycine protein sequences. Intermediate frequency is found in asparagine residues followed by a polar amino acid with a relatively small side chain, for instance serine, threonine, or aspartic acid. Low frequency is found in asparagine residues followed by a hydrophobic amino acid with a bulky side chain. There is different rates of reactivity, as steric hindrance by the bulky side chain of the N+1 amino acid may limit accessibility of the reactive nitrogen N ion to the asparagine side chain amide, thereby reducing the rate of reactivity. Exposure to alkaline pH also favors deamidation. Exposure to alkaline pH results in an increased rate of succinimide formation due to greater deprotonation of the peptide bond nitrogen at higher pH values. I am going to talk about the analysis of deamidation using protein L-isoaspartyl D-aspartyl carboxyl methyltransferase, which is also known as PIMT, followed by the reverse phase liquid chromatography, which is also known as RPLC, followed by the electrostatic repulsion hydrophilic interaction chromatography, which is also known as ERLIC, and followed by Fourier transform mass spectrometry electron capture dissociation, which is otherwise known as FTMS ECD. PIMT is a useful tool for isoaspartyl analysis. Deamidation of asparagine residues results in the formation of the intermediate succinimide. Upon spontaneous hydrolysis, L-isoaspartate linkages are produced. The enzyme protein isoaspartyl ethyl transferase can be used to specifically detect the presence of isoaspartic acid residues in the target protein. PIMT catalyzed transfer of the methyl group from S-adenosyl L-methionine SAM to isoaspartic acid at the free alpha carboxyl position. Spontaneous decomposition of the resulting methyl ester will cause formation of a cyclic succinimide releasing 3H-methanol as a byproduct, which can be measured by scintillation counting. The spontaneous opening of the succinimidyl ring will result in formation of either the normal L-aspartate peptide or the initial L-isoaspartyl containing peptide. Repeated methylation cycles allow quantitative conversion, which results in the final substrate, D-aspartate. An alternative technique is the use of radioactive SAM, which is based on an ion pairing RPC method. This rapid and reliable procedure specifically detects and quantifies S-adenosyl homocysteine, which is a byproduct of the reaction between the enzyme protein PIMT, SAM, and isoaspartate residues. D-aspartate containing peptides are also substrates for PIMT. An alternative technique is the use of radioactive SAM, which is known as S-adenosyl L-methionine, based on an ion pairing reverse phase chromatography method. This rapid and reliable procedure specifically detects and quantifies S-adenosyl homocysteine, which is a byproduct of the reaction between the enzyme protein PIMT, SAM, and isoaspartic residues. D-aspartate containing residues are also substrates for PIMT. RPLC is another method for detecting deamidation. Deamidated proteins are often digested enzymatically to smaller pieces to localize the deamidation site using trypsin. C18 non-polar stationary phase can be used. ACN can be used as the polar stationary phase. Retention degree is based on hydrophobicity. The proteins are eluted in the order of increasing hydrophobicity. This results in a pattern of peaks or fingerprint diagnostic for a particular protein and used to monitor product identity and purity. RPLC can be used to determine the ratio of products. The disadvantage of this approach is that isomers aspartate or isoaspartate peptides separation can be challenging. The slight structural differences between these two residues in peptides can make their separation challenging, so shallow gradients and long runtimes are often necessary for baseline separation. For instance, a gradient of 15 to 19% organic mobile phase in 60 minutes was required to achieve baseline separation of isoaspartate or aspartate peptides from a tryptic digest. Thus, many HPLC runs are often required before adequate separation is achieved. To solve this problem, Ehrlich can be performed after RPLC. Ehrlich is also known as electrostatic repulsion hydrophilic interaction chromatography. This is a mixed mode chromatographic technique which combines the use of electrostatic repulsion and hydrophilic interaction. These two forces will act during separation. In RPLC Ehrlich, there are two dimensions of liquid chromatography. The first dimension is RPLC, where peptides are separated by hydrophobicity. The second dimension is Ehrlich, where peptides elute in order of their isoelectric points and polarity. There will be a uniform distribution of peptides among the collected fractions, with peptides eluting in order of decreasing isoelectric points and increasing hydrophilicity. It is promising for the study of peptide deamidation to separate the isoforms asparagine, aspartate, and isoaspartate. Once separation is completed, FTMS ECD can be used to differentiate the isomers. ECD, which is otherwise known as electron capture dissociation, is a sequencing tool that involves the reaction of electrons with gas phase multiply charged peptide and protein cations. The captured electron initiates N-C bond cleavage, producing C and Z type fragment ions, which are known as the N and C terminal fragments respectively, and can also preserve post-translational modifications linked to the fragments. ECD can also cleave bonds in amino acid side chains, such as the CC bond. Fourier transform mass spectrometry is based on the circular movement of charged particles in a strong magnetic field, which is otherwise known as the cyclotron movement. The cyclotron frequency depends directly on the mass-to-charge ratio of the ions. The periodic movement of ion packets is recorded and converted to a frequency spectrum with a Fourier transform, which is converted to a mass spectrum after calibration. The major advantage of using this mass spectrometry is its ability to resolve interferences. For example, a peak of mass 800 daltons can theoretically be distinguished from a peak at mass 800.001 daltons. These performances make the technique highly valuable for multi-residue analysis. The other advantage of this spectrometry is that new fragmentation techniques can be used in conjunction with it, such as infrared laser activation or electron capture dissociation, which is mentioned earlier. These two techniques can be used in combination to fragment very large molecules such as whole proteins. FDMS ECD can differentiate isoaspartate from aspartate residues in synthetic peptides without the need for control peptides. It uses diagnostic ions unique to each form to indicate the presence and position of isoaspartate residues and aspartate residues, showing that fragmentation patterns in peptides from proteins will demonstrate the applicability of this method for differentiating the two forms of aspartate acid in whole proteins. Analysis of deaminated peptides from proteins with HPLC and ECD demonstrated that the combined technique easily differentiates and determines the relative abundance of products.
Robinson hypothesized that deamidation of glutaminyl and asparagine residues in peptides serve as molecular timers of biological events such as protein turnover, development, and aging. Human aging is associated with the deterioration of long-lived proteins. Gradual cumulative modifications to the lifelong proteins have been shown to have implications in Alzheimer's disease, cataract formation, celiac disease, and cancer. In this presentation, we are going to talk about current studies on the relationship between deamidation and vascular dementia. Vascular dementia is the second most common dementia after Alzheimer's disease. Dementia is associated with memory loss, difficulties with thinking, problem solving, and language. As the life expectancy in Singapore increases, prevalence of dementia is projected to increase to more than 50,000 by 2020, thus making it a target for treatment and therapy. Ion channel proteins, such as the sodium potassium ATPase, is involved in ion transport across the membrane in the neurons. The protein activity is associated with the formation and persistence of long-term potential in neurons important for synaptic plasticity and memory formation. Deamidation of ion channel proteins also interferes with sodium-potassium pumping and affects signal transduction in the neurons. To study deamidation of proteins in vascular dementia, clinical samples of healthy and dementia subjects were acquired, and the researchers used a discovery approach to identify targets having roles in vascular dementia and subject them to further studies in the validation approach. One key technique to highlight in this paper is the use of eye-track labeling to quantify and compare the protein expression level between healthy and dementia subjects. Briefly, the quantification is done through incorporation of stable isotopes, and the isobaric tags can be used for relative and absolute quantification of the proteome. Besides that, eye-track can also be used in biomarker discovery, time course, and drug treatment studies. Utilizing eye-track, it is found that voltage-dependent and ion-selective channel proteins were downregulated compared to control subjects of the same age. Proteins such as sodium potassium ATPase subunits were upregulated in vascular dementia patients. These findings suggest that the dysregulated ion channel proteins. Bioinformatics analysis also revealed significantly higher deamidation in ion channel proteins in vascular dementia patients. This slide shows the structure of two conformations of the sodium-potassium ATPase. The asparagine and glutamine residues near the catalytic sites, highlighted in red, are essential for magnesium ion binding. Thus, they play a major role in maintaining activity of sodium-potassium ATPase. The deaminated asparagine and glutamine residues close to the catalytic sites are found to cause defects in phosphorylation and dephosphorylation mechanism, and thus affecting the ATP hydrolysis and energy supply for the ATP-coupled ion transport. Without this process, proper functioning of the nervous system will be affected. Interestingly, it is also found that PIMT is deamidated in vascular dementia. It has been mentioned before that PIMT repairs abnormal proteins. Therefore, the amidation of this enzyme will cause defect in protein repairing function and accumulation of isoaspartate. This may prove to be an interesting target for further studies. To conclude, we saw that mass spectrometry is essential for discovery and characterization of modified proteins. In addition, when amide residue is present in a peptide or protein, it is definitely a miniature molecular clock that runs at a predetermined rate and changes the peptide or protein structure in specific ways. We also saw that deamidation is a biologically relevant phenomenon in human proteins, as it has implications in various diseases. Finally, deamidation may be potential targets for drugs and therapy to combat degenerative diseases. These are the references we used for our presentation. Thank you.